For the Evos FL fluorescence microscope. This is a new device within the McMaster Biophotonics facility, and it has fundamentally different capabilities than some of the other scopes that are in here within the facility. This is really a device about high content analysis, image stitching, both color and fluorescence imaging, histology, and the ability to look at plates of 384/96 wells at the level of fluorescence. It's also an excellent microscope for time course microscopy because it is a fully incubated microscope with both CO2 and temperature control. So the difference about this microscope versus other microscopes rests in the light source. And here they are. This is a typical cube, one of 4 of which can fit into the Evos at any given time. The cube consists of a single frequency LED light on the top. That's under this copper heat sink up here, which will provide one frequency of light through a dichroic filter and a filter cube to the sample. Then the emitted light comes back through the filter cube and gets sent to the camera. So unlike a regular fluorescence microscope that has one light source and the light is filtered for the different frequencies of light, in this scope, each fluorophore will have its own unique cube. And while there's a position for 4 separate cubes in the microscope, we can choose from a library of many cubes available from the manufacturer. So as a result of that, this is a slightly different microscope than most. In addition, the other major difference is this scope is controlled almost entirely through the screen and by touch screen control. So there's no eyepieces as you would expect in a regular microscope. To turn it on, there's 2 switches. There's one switch on the actual scope base itself, and then there's the computer control. The computer is also not like typical microscopes in that no data can be written to the computer. It must be written to an external solid state or hard drive that's plugged in through a USB 3 port. So on the top of the computer is a USB 3 multi-port tap where you can plug in either a stick or a hard drive and take your data with you immediately. So none of the data is actually stored on this device during data acquisition. The interface is a touchscreen Windows 7 Professional. And it works like any typical computer. So once the scope is on, we're going to turn on the one and only icon that should be on the desktop to start the Evos FL Auto. If the scope is not on, it will give you a warning telling you that the scope isn't on and that you need to turn it on. And what you're going to see once it boots up is that it has a relatively straightforward user interface that actually makes simple acquisition pretty simple compared to most microscopes. So once it starts up, what you're going to see here is a graphic representation of the type of stage insert that we have on the scope, and an indicator of the approximate position of the objective on this stage. So what I'm going to do is change the current stage insert, which here is designed for 4 30mm tissue culture dishes, and I'm going to adjust it to a dual slide holder. That's in the software. All right, so on the microscope stage, this is an incubated and sealed unit. Now, it doesn't have to have the gas and temperature control on for imaging. In this case, for the sake of this demo, we're just going to be looking at a fluorescent slide that's stained with multiple dyes. The holder for the slide fits in to this milled aluminum unit. We have several potential holders. For unincubated tissue culture dishes, we have this 4-well holder for 30mm dishes. For incubated dishes, we have these special 30mm holders, which are actually designed to hold down the tissue culture chamber and provide a seal in the incubation gases and temperature. But for the sake of this demo, we're going to look at this dual slide holder, which I programmed into the software, and we're just going to gently put that into place on the microscope, and you can see that in place we have our 20x air objective located in the center position. I'm going to just put on the prepared slide, which has been cleaned because we're using an air objective for this demo. And we're going to drop that into place. And that's basically it. From this point forward, you really don't need to touch the microscope at all. So we can put on the top of this chamber, which is this plastic device that's held on by magnets. And there's a polarized stage to prevent any ambient light from getting in, but if you want, you can seal off the light completely with the use of this cover. All right, so now when it comes time for acquisition of the data, we have a series of options here. Here's our program slide holder, and this is telling us the approximate location of our objective. And this can be adjusted either directly through the touchscreen interface or by using the mouse controller. We also have our choice of objectives. And currently on the scope, we have a 10x air objective, a 40x air objective, a 20x plan apochromat air objective, and a 60x oil plan apochromat objective. The 60x oil is the highest resolution and highest sensitivity objective that we have, and then the highest magnification. Although you'll see for many applications, the 20x plan apo is in fact an excellent objective for this use. We're now going to choose over here on this row. We can see what the various different cubes are for the various frequencies. What we have in place now is a Texas red cube, a transmitted light setting, a YFP cube, a DAPI or hushed cube. So we're just going to switch this over to the YFP cube, which is just a green fluorophore, and we're going to turn on the fluorescence light, and we're going to look for our image, which means we're going to have to do a coarse focus adjustment. Now, the focus adjustment can be done by your finger by dragging the slider, which I don't recommend because it's not sensitive enough. You can grab it using the cursor on the mouse and sliding it, which is also can be not very sensitive, or you can roll the wheel on the mouse to give you a pretty approximate coarse focus, and then you can go over to fine focus to get the absolute ideal focal plane for your image. And here we can see a nice green image appearing on these cells. Once we've done this, we're going to now set what's called the focus nominal. And what that's going to do is tell the microscope the approximate range in which your sample exists in order for autofocus to occur if we want to do that. And what we can then do is look across the channels and adjust the light settings. So there's a simple and there's a more advanced light setting measure. The simple light setting measure is just this slider that will increase the amount of light that goes into the system. And as you can see, what happens is once we increase the light too much beyond a certain threshold, we'll get red pixels appearing, which says we're out of the linear range of the electron multiplied CCD camera that's on this device. So we're going to turn down the light to get us within the linear range so that we can do quantitative imaging. And we're going to switch between the other channels. So for example, on Texas red, now our focus is already set because we did it on the one channel, and we're just going to turn up the light on that to get and avoid the presence of any red pixels. And we're also going to do this on the DAPI channel. Okay. If we press the light and camera tab, we have an optional mode to adjust the camera, which is the preferred mode, which you can click on here. And what that'll do is actually give you control of the light level of the LED, plus the exposure level of the camera and the gain level of the camera, because it is an EMCCD camera. So we're going to set the light at a certain level, we're going to increase our exposure, and we're going to increase our gain appropriately. We'll switch over to the green image. All right, so what this allows you to play with is examples of cases where you're worried about photo bleaching of your sample. So if you are worried about photo bleaching of your sample, what you probably want to do is reduce the exposure time, reduce the light levels, and increase the gain as much as possible. So you can see now we've got a nice image with a fraction of the amount of light as before and a very short exposure time, which means for every exposure we minimize our photo bleaching. However, what you may be able to see, but what you can see in person, is as you push up that gain to a maximum here of 24 decibels, you're going to see the increased amount of noise in the image. So if you are interested in quality and not so interested in photo bleaching, then you can reduce the gain on the image, increase the exposure time, and increase the light amount. And with very little gain, you will probably not see any visible amount of noise on the camera for optimal imaging. And you can adjust these settings independently across all three of the channels. So I'm going to turn down the light here, and I'm going to leave the light setting there. Now, you've got a pretty wide image of view here on your sample, and you can always tell where you are approximately on the slide or the tissue culture dish by this blue dot, and you have the option now of, if you want to, move around on the dish. Okay, so we can now look around on the dish by simply dragging and moving the sample by finger and moving to another part of the dish. And if you want to adjust focus, there's an option of pressing the Auto Fine Focus feature. And what that's going to do is quickly scan through the Z-stack plane near the image and try and achieve the best possible focus. And as you can see, for the most part, the automatic focus is quite accurate and quite impressive. Okay. Decide to capture an image, we can decide which channels we want in that image. And the way we do that is up here in these tabs in the upper right-hand corner. So under Settings, we want to capture these channels on the Capture All command. And what I've shown here is I want to capture Texas Red, the YFP, which is just green, and the DAPI channel. And you can specify the type of format that you want as well. You can also specify the pseudo color that you want for each one of the channel images. For example, in this lab we prefer to use magenta as the red color rather than actual red, because that's what's preferred by the press for publication. So back to the Capture tab, we're now going to press Capture All, which will give us Texas Red, and then YFP, and then DAPI. And that'll give us our final image. Once we have our image then on screen, we can magnify in on the image and look around using pinch to zoom as you would on a tablet or a phone. And that'll give you an idea of exactly what kind of quality of image that. If you do press Capture, you then have to go over to the Save bar to actually save the image. And then you have a choice of the bit depth that you want to save it in, whether it's just as a qualitative image by 8-bit, or if you want to quantify it down at 16-bit. I recommend the use of the TIFF file format because it contains all the data, and you can save it. If it's one channel, you can save it as a monochrome image. If it's multiple channels, you'll probably want to save it as a color image, and then you'll press Save Image. And it'll ask you to for the location of where you're going to save it on the hard disk or thumb drive that you've plugged into the USB port. All right, for simple image capture on any given channel of your image, you can just use these buttons to flip between the various light paths through the cubes. You've set your exposure rates up here, and they're going to be individually set for each channel. If you have an image that you're happy with and want to capture it, you'll just press Capture. The image is now captured, but it has to be saved. So you go over to the Save tab, and now you'll see the location to where your file is going to be saved, and you have your options of how you're going to save the image, either as a quantitative monochrome, which is going to be in 16-bit image, or you can do a custom setting. For example, you can do a monochrome but 8-bit. I recommend the use of the TIFF file format because this is not compressed in any way and gives you all of the actual data. And you have the options of also including image annotations if you're going to put them on and saving underlying images. So you're going to save it on its location onto the hard disk or thumb drive that you've plugged in. If you want to do multi-capture imaging, then we review again in the settings what images we're interested in capturing. So let's say we want red, green, and blue on the image, and we now press the Capture All button. Okay, with the Capture All button, you're going to see other options come up once the image has been taken. And again, you have the button down here to save the image. And there's various options you can have for saving that image in terms of the file format that you want and the location. Once the image has been captured, you also have the ability to open it up and look at it with higher magnification, just using pinch to zoom gestures on the screen, as there is a touch screen control. All right, so the other option is to just look at a transmitted light image without fluorescence. Now for this, you're going to have to remove the cover on the scope to allow the light through, and you're going to have to do your adjustments. So you're going to need a very short exposure, very little light, and you should see a bright field image of interest, which you can capture on as well. In these settings, what we have are in fact 3 cubes that are in place for red, green, and blue, and we have in fact no cube to provide for a transmitted light image. But that transmitted light image is just bright field. It doesn't contain any optics to do phase or differential interference contrast imaging. For that type of imaging, you can switch over to using the color camera and adjust our light levels, and we're going to remove the polarized top, which has a filter effect that can give it kind of a yellow image. We're going to turn down the light until we get to the range where we're getting decent color imaging. Okay, this is a very flat fluorescent sample, so there's not much to see here. But this is just to highlight the fact that this scope does have a color camera, so it can do histology slides. Okay?
Yep.
All right, so this device has a very good automated stage. It's a very precise stage, and it also has autofocus features, which means that you can actually scan a very large area of your slide and conduct image stitching to give you a very large image of a very large area under your slide. And if you want, as large as you want. It's basically limited by the size of the hard drive that you've plugged into the microscope. So I'm going to give you an example of how to scan an image slide. The first thing we're going to do is look at our sample. We're going to turn on our power. We're going to grab an area of interest, something nice to look at. We're going to do our auto fine focus, which we can do because we've done capture focus nominal to give us our proper range, and you're going to see that the focus is turned into a nice sharp image. We have the correct exposure that we like, and now we're going to go over to the scan tab, and this is going to follow you through a routine on how to generate your scan. So we'll click a new routine, create a new routine, and we're going to give it a name. We're going to choose what kind of plate we're looking at, and you'll see that there's many options on the type of plates you can look at. 6, 12, 24, 96, all the way up to 384 wells, all branded by the proper manufacturer as well, which is really important. In this case, we're going to be looking at a slide sample on the slide holder. You can then choose which objective you want to use, which camera you want to use, either the monochrome for fluorescence or the color camera for color bright field imaging, and then which channels you want to image. For the sake of simplicity, I'm going to stick to a 20x objective, a monochrome fluorescence camera on the green channel with the YFPQ. You have a few options here. You can save the individual lighting channel images separately, which requires more disk space. And you can show this page again when you actually run the scan if you want, but we're not going to click those. You can decide how you'd like to scan the area. Typically people are going to choose something like an area rectangle, but you also have the options of choosing beacons, which are positions on various positions anywhere located at the bottom of a tissue culture dish, which we'll run through later. So what you're going to get then is an image of your slide and the current location of your objective, and what you're going to be able to do is set the area in which you're going to scan your image. And you're going to be able to change the position of your scan area as well as the size of your scan area by adjusting with these buttons. So if I want to make the scan area a little bit wider and a little bit taller, I now have a bigger area to scan. In addition, I could also decide to scan onto the second slide, but in this case we only have a sample in the one slide. We press next. The next option that you have is as you move across the slide, you want to have the option of being able to adjust the autofocus or not. If you're worried about bleaching of your sample, you don't want this option. If you are not worried about that, then you go ahead and do the auto fine focus operation. And then you have the option of how much coverage you would like of your slide area. You could literally image the entire slide with overlapping images that will be properly stitched together in full coverage, or you can select sub-regions of the area of interest, and you can define these as well as you want. The question of why you would use one or the other is a matter of whether you're interested in seeing absolutely every cell in the area, or if you only want to get representative images from approximately that area on the dish or the slide. And for the sake of this demo, we're going to look at full coverage on this area. Press next. It'll now give you the options to adjust the focus and the lighting situations, but in this case we're fine. Press next. You're going to choose a folder. We're going to make a new folder in our case. And we're going to save it into there. We're going to press next. And what we're going to see is a summary of what's about to happen. For this area, we're going to need to image 234 separate images we have on one slide, and it'll take approximately 551 megabytes of data. This is really good because what it tells you is that it won't actually let you do an experiment if you don't have the room to save it on your hard drive. And then it's a simple matter of pressing start. And this is going to take place, and it's going to take a while. So you have two options here. As you can see now, as it's starting to capture the image, it's going to do a focus adjustment and capture the image, and then move on to the next area of the slide. And we're essentially building a bigger and bigger tile in the entire slide image area. Another option you have with this remote is to turn on the plasma screen TV, which mirrors the display on screen here. So you can either watch it from a distance or just peek into the room during the procedure just to make sure everything's going okay without actually having to sit at the scope. This is also good if you want to work with someone else so they can have a view of the entire mirrored display image on here. So what this tells you then is that we're on image now 18 of 234, 56 seconds has elapsed, and this total time is going to take 16 minutes. And if you're looking at the stitching as it's occurring and you're seeing that everything appears to be fully in focus and there's no drift and there's no issues, there's actually no reason to sit here and stare at the scope, and you have 14 minutes to go grab a coffee. All right, the next experiment we're going to program into the Evos FL is a time-lapse experiment. So this can be done in two ways. We can either look at one area of the dish in a time-lapse experiment for as long as we want, or we can actually capture multiple areas of the dish over a period of time. So right next to the scan button is the time-lapse button along the top. We're going to create a new routine similarly to what we did for the scan. And we're going to have our options now of our objective type, the camera type that we're going to use, the channels that we're interested in, and we're going to have the ability to auto-find your sample in that channel and also do auto-find focus in that channel. We also have an option here to do a Z-stack, which we'll explain after doing this module. The next part of this programming is the concept of beacons. Beacons are basically points on the dish that you want to image over time. If you just imaged, let's say you're very interested in this one cell here, you can set your beacon to that position in the crosshairs and just say add beacon, and you now have one beacon, and you can go forth and do the experiment where you're going to do a certain number of images at a certain number of times. On the other hand, if you're interested in looking at several interesting cells that are not all in one field of view, you have the option of moving around on the dish, taking another cell in your crosshairs and assigning that as a second beacon. And you can go into a completely different part of the dish and assign yet a third beacon. And you can do this for up to as many beacons as you want. What this allows you to do is at the start of the experiment, scan over the entire surface of your tissue culture dish, find all your cells of interest, for example, at a certain point in the cell cycle, and start your observations. And at each time point, you're going to go to each beacon position in the discs and take a picture. And at the end of the experiment, what you'll end up with is multiple data stacks representing each position of the plate for that experiment. Okay. So in this case, we're looking at 3 beacons, which we programmed in for location. We're going to look at one channel, and our focus is fine, so we're going to press next. What type of file format would you like to use? I prefer TIFF. You have the option of saving individual channels. You can create also an option of a video for each beacon. Alternatively, you can just take those stack of TIFFs and using simple software available online, freeware, you can generate movies pretty easily with programs such as ImageJ. Okay, so what'll happen now is you might get this warning here that says collect samples, and it states the software is unsure that it can collect the time-lapse samples in the time available. Would you like to run the time-lapse as fast as possible anyway? What the software is telling you is that your interval of 1 minute may not give it enough time to move between the different positions on the stage. So the easiest way to fix that is go back and choose your beacons, fewer number of beacons closer together, or increase the time between your captures. So let's say you do it every 2 minutes, for example. So now that we've capture every 2 minutes, I should give it plenty of time to move in between. And we press next, and you can see that warning does not come up. And then it says you finished setting up scene 1. And then press next to say the next time-lapse routine. So the idea of stacking scenes subsequent to this can be done for something, for example, drug treatment of cells, where you want a certain period of time where you're watching normal cell growth in the absence of any foreign agents, and then you want to start a second scene where you're going to add in your drug of interest and watch that drug do its activity, and you can add a third scene where you now wash out the drug and watch it return back to normal. In this case, we're only going to be looking at one scene. It's going to ask for a position to save it to, and you then have the option of start. And once again, you're going to get a summary. and you're going to see that you're at scene 1 of 1 and you're at beacon 1 of 3. So it's going to autofocus. It's done a Z stack through the image. It's going to choose its auto find focus, and then it's going to move over to the second beacon. There's your capture. It's now moved over to the second beacon, and again it's doing the autofocus, and it's going to do its capture. Then it's going to jump to the third beacon, and it'll do this for the number of shots that you require in a certain interval for a certain period of time. The good thing about the software is it will do all the math ahead of time to tell you if you have enough hard drive space to do the actual experiment. And if you don't, you're going to have to plug in a bigger hard drive. That's why I would recommend that your lab purchase some of these convenient portable 1 terabyte hard drives for this type of work, because that'll give you more than enough space to do very, very long experiments and do very, very large captures. Just for the sake of this demo, I'm going to press finish on this, which is stop up here. I'm going to just discard. So basically you can back out of the experiment if you want. And we're going to go back to the home screen here and look at our image. So we're going to talk about how to set up a live cell incubated experiment. This can either be temperature or both temperature and CO2 incubation. The incubator that provides both the heat and the carbon dioxide mix is this white box in behind the Evos FL. And it has a toggle switch on the back to turn it on. This is the Evos FL incubation chamber that provides the heated, humidified, and proper CO2 levels to the incubated stage on the Evos FL. The unit is turned on through a power switch at the back of the device right here. Prior to an experiment, it's essential to make sure that there's enough water in the incubation tank under this cover for the length of the experiment. Generally speaking, 1 tank of water will last quite a long time, in excess of 24 hours. But it's just important to check it before you start your experiment for your live cell incubation. To open it, you just pull on this latch, pull it up, and you can pull out the tank. And the tank is just filled with water. And what you will see is that there's an important sticker right here indicating the maximum fill level on the tank. It is not the top of the tank. The maximum fill level is in fact this line right down here, which is not very high. It needs this head space of air above the water to make sure that you have proper mixing for the humidity and gas mixture. So just make sure that the water is filled to that approximate level, and you should be fine for at least 24 hours. Just using tap water, and the tank goes back in and just sits in like that without any effort, and the top just clicks right back down. Now what you want to do is make sure that that water is warmed up to the right temperature so it will properly adjust for the proper humidity levels prior to an experiment. And that means you're going to need at least 90 minutes to turn this on ahead of time to make sure that it's come up to temperature. That device has to be turned on, and the scope base has to be turned on before you start the software. Once those are on, you can now launch the software. Over here at the gas tanks, we now have to turn on the gas. So in both cases, we're looking at an air tank and a CO2 tank. And what we're going to do is just open up these thumb screws to release the gas flow into the gas mixer. And that's pretty much it. One thing you want to keep an eye out is the total pressure that's in the tank before the experiment, and if that pressure is low, to make sure that you contact someone back in the truant lab to replace these tanks. Okay, so typically the incubation system should be turned on well in advance of doing the experiment if you want to set up for incubation. And the reason for that is there's a heating element both inside of the stage and at the incubation chamber that must be brought up to temperature. In addition, there's a requirement for humidity, and there's a water tank within the incubation chamber that needs to be filled to a minimal level to make sure that the sample is not going to dry out over time as it's being heated up. All right, experiment. We've set our focus and our exposure levels on our sample. We're then going to go over to the time-lapse tab, and we're going to start clicking left to right on the Incubate tab, and you're going to click on Enable Environmental Chamber. And now that you've done that, you have the option of setting your temperature and your CO2 level. And you have to make sure that the lid is on the device and that it's properly seated in position so you get a good seal. And you also have to make sure that you have the appropriate vessel holder to provide a seal for both temperature and gas control. And what you'll see here is currently, because we just turned this on, the values for temperature, humidity, and carbon dioxide will be shown as adjusting with their targets present here, and that should disappear after a short period of time. We then work left to right and click on the routines. And now we're going to create a new routine, very similar to the other routines that we've done. And again, we have all of our options for our objective, our camera, our channels, auto-find sample, auto-find focus. And again, we can choose, similar to before, our beacons. We can add our various beacons around the dish. In this case, we're going to have just one beacon. The file format, and the number of scenes. Now, the scenes in the case of incubation is the ability to stack different experimental conditions of incubation. So for example, you could go from 37 degrees down to room temperature, back up to 37 degrees again. You could go from 5% CO2 to no CO2 to back up to 5% CO2 in a third scene. In this case, we're just doing one scene. And what we're going to do is going to do a short experiment, capture images every 1 minute. We only have one beacon, so there won't be any moving around on the dish. And here's our environmental conditions. And
make sure that it actually has CO2 clicked with that. CO2 is never defaulted clicked.
Oh yeah, so if you're using the environmental chamber, make sure that the CO2 level is adjusted at 5%. You'll see a grayed out area here for oxygen control level. We don't have that capability on this yet. So we've now finished setting up scene one, and then next is going to decide where the time-lapse routine. There's the folder that's going to save it to, and you can hit start of the experiment. And here we can see we're in the first scene, and we've imaged the first beacon position. And we have 5 minutes remaining, and the disk space is going to be 124 gigabytes used of 340 gigabytes remaining. So we can carry out this long incubated experiment for as many hours as we think we're going to need. So for the sake of this demo, I'm just going to stop this and discard it. Yeah, currently in the software, there's no way to actually review the images already taken. It's only at the end of the experiment. Hopefully that's something that's going to be addressed soon by the software engineers at the company. It's very similar to our deconvolution microscope that we have in the MBF. So we're just going to set our fine focus here. Auto fine. And we're going to do a Z stacking experiment where we're going to do a Z stack through space on our cells of interest. So I'm just going to adjust the fine focus to the proper point, and we're going to press now the Z stack, and I'm going to do this actually in 2 images, 2 channels. That's fine. I'm going to press the Z stack button, and now what it's going to do is tell us the positions that we're looking at. So we're interested in 2 channels here, YFP and Texas Red. The current Z position is right here. What we're going to do is we're going to set the top and we're going to set the bottom. So to set the top, we're just going to move the fine focus slider up till we've reached the top of the sample and say set top. Then we're going to focus down through the correct focal point and then out of focus again through the bottom, set bottom, and that's going to now give it our range of slices. It's going to automatically compute the optimal Z stack parameters, so the optimal distance between the Z so you're not losing any light information. And it's figured that out as 0.366 micrometers for the objective that we're using, which means 44 images. Now that's a lot of images. There may be a situation where you're worried about photo bleaching, in which case you want to override this and go to manually enter your step size, where you could, for example, decide that you only want 20 images spaced twice as far apart. For now, we'll leave it on automatic, and you have the options of saving individual images and the flattened image, which means all of the Z images flattened down into one stack, and it's going to ask you again for a location of where to save it. And again, you press start. And what we're going to see is the start of acquisition. So first it's going to go through the red channel and march down through the exposures through the Z plane. And you can see in the summary here there's 88 images across the 2 channels, 44 in each. If you look at carefully, you can see the progress bar is going along, and if you look very carefully at the sample, either here or on the plasma screen, you can see that it's slowly coming into focus as it comes to the correct focal plane, and then goes out of focus again as it goes through to the bottom of the image. If you choose the Auto Z Stack feature, this will give you a Z stack that has the proper spacing for image deconvolution. And you can take that Z stack and bring it into other software that can deconvolve that image to give you the highest possible resolution for that frequency of light using standard resolution methods. Okay, so we're about halfway through on our progress bar, and you can see that we're now switching over to the green channel, and with exactly the same parameters for the number of steps and the exact same focal points as we did through the red channel. Now, once you've done this, you can now start mixing and matching and do a time-lapse experiment where at each time point you can do a Z stack image in multiple colors to do what's called 4-dimensional or multi-dimensional analysis. So you can actually gain a 3D image from every time point during a time-lapse experiment, and then look at multiple proteins in the different fluorescence channels. And you can see the green is going a little bit faster because the exposure is faster and the sample's brighter in this. And then what we can see is the flattened image at the end of the experiment, which has all of the stacks put together to generate the final image. If you have a very dim image, flattening a Z stack is one way to get the maximal amount of information out of your image, because what this does is it sums all the light coming out of your sample to put it into one image. So it's quantitative, and it's also the way to see very, very dim samples in unusual situations. And once it's done, we're just going to press finish, and it's going to tell us, it's going to ask us where to save the image.